Hello?
Hydroxychloroquine.
Oh, okay. So, what target is the drug aiming for in the body? And what sort of human trials have been done to confirm that the drug indeed actually works as you think?
I feel good about it.
Oh, that's great that you trust it. That's really good. That's where it all starts. But for the rest of the community, the rest of the world to trust it and to accept that it's actually going to do what you say it does, to want to spend money on it, not waste their money buying the drug, or worse, it could be dangerous to their bodies. You're going to have to show that you've done some tests, you've measured the safety of it and make sure that it's doing what you think it is doing. Yeah, there's some studies that you should have done. Hi, today we're going to be talking about the process of discovering drugs for the treatment of disease. In order to do that, we're going to look at three main components of the process, which is identifying a target identification, then high throughput screening to identify a small molecule that can be progressed on to make a drug, and then the final step in the process is looking at making the molecule more drug-like. And you understand what that means as we go through the process. So let's get started. A background. Who gets involved in drug discovery? Who are the key players that may be interested in looking at discovering new drugs? So as you all may guess, pharmaceutical companies and industries were for the longest time the main players, but more and more research and academic institutions are very much involved in this space too. So you start off then in this process with what's called a target identification. This is where you identify a target in the disease phenotype. Phenotype just means what it looks like. To do this, you check hundreds of thousands of what are called small molecule compounds for their ability to alter what the disease looks like. The modern approach, the current approach, is to more focus on a target. We will be clarifying the drug discovery process. We will learn that a compound does not become a drug until it's gone through several steps and it passes those processes. That's then and only then will a compound become a drug. It needs to be made drug-like. The compound that you find in the drug discovery process needs to be made drug-like, and we will cover what that means. It needs to be tested in preclinical trials, so usually animal models. Well, definitely animal models, and then it needs to be ultimately tested in humans, in human volunteers, in clinical trials before it can be released. And if you have a look at the process, a lot of compounds, the majority don't get to this stage. So let's have a big picture view of the drug discovery process. So the best thing to do that is to use an example: malaria. If I want to intervene with malaria as a disease, what are the things that I need to consider? I first need to understand the symptom that identifies malaria. I need to understand the unique symptoms that identify the disease. I need to understand what causes the symptoms, maybe what is called the etiological agent. So you need to understand what, the organism that causes it, whether it be viral, bacterial, or whatever component is causing the disease. It's good to get a grasp of that. We know that it's, in the case of mosquito, it's a parasite that's inside the mosquito. It's not actually the mosquito. So, the system needs to be studied to actually identify the real reason, the real cause. And then you have to look at how does this parasite actually interact with the body to cause the problems, to cause the disease that it does. What does the parasite, once it's inside the body, what does it need in order to be able to survive inside the body? And once we know that, then we can attack what it needs. And that's essentially what you're doing in treating it via a drug. The drug will target, in this case, the parasite need to survive. If we can stop that, then obviously we can intervene in the disease. So if we can look for structural entities or compounds that interact with that parasite and stop parasite's ability to cause disease inside the body. I called it structural entities because at this point in the drug discovery process, it's not a drug. It's a chemical structure that interacts. Well, the thing is, the chances of finding a molecule that interacts with the disease, in this case a parasite, that gives it an outcome that we desire is very, very low. So you can't just look at a handful of compounds and come out with something successful. Typically hundreds of thousands, not hundreds, 2,000, but hundreds of thousands of compounds, of structural motifs are checked. And this is what's called screening. And so the main part of drug discovery, a crucial component of it, is the high throughput screening. The more readily recognized is the high throughput screening component, which we'll be looking at. So you check these compounds, these structures, chemical entities against your target. In drug discovery, usually, the target identification is a crucial part of it in the high throughput screening lab. That's not what the high throughput screening lab's job is. Their job is to screen the small molecules with your target. So you usually have to have identified your target, validated the target, as we will discuss, and then you go into the screening to find compounds that's interacting with your target. And it's called high throughput because it's not just a handful of compounds which you could have just done on your lab bench. It's hundreds of thousands of compounds. So the system is usually automated. You're using robotics to be able to process that many compounds. So if any of the compounds or small molecule structures are noticed or flagged, as you see, this is called hits, in that they are interacting with the target of the disease in a way that is promising. Then it is studied further with the hope and aim of making it into a drug. Also, how do chemists even know where to start? Like, how do they know what structures or compounds? Well, the best way to do something is to just copy an existing one, right? So you know that in nature, there's some compounds or chemicals or structures that we know kill bacteria, or we know kill fungi. So you can sort of study those and get an idea of what their structure is like, and then that can give you like a starting point to creating new structures. And the word novel comes up again, a novel structure that doesn't exist. So you can sort of come using that scaffold, create more and more and more and more, hundreds and thousands of these that in a new disease setting can be checked to see if it's interacting with these. Then you need an assay. An assay simply means an experimental setup that allows you to check if any of the compounds can interact with your target. You need to miniaturize the experiments. You need to go from the standard experiment volume, such as an experiment that you would have done in an Eppendorf tube or in a test tube or use larger format. You need to miniaturize it. What do we mean by miniaturize it? Well, we're going to be checking hundreds and thousands of compounds. This is going to be expensive. Reagents are expensive. Your reporter, this experimental setup, all the components going to will be expensive. And since we're checking hundreds of thousands of compounds, we need to check it small, reduce it to the smallest volume that still allows us to detect differences. That still allows us to have a solid experiment, what is called a robust experiment. The statistics and measurements you can use to check that you do have a robust experiment. We will get into that. We have an assay, we have a target, we have an assay, an experiment that's going to let us probe the system to check that our target is being interacted with in a way to be able to detect it. That's your assay. So now let's go to the heart of the matter. Well, at least what's readily associated with the process. So that comes to the high throughput side. So the high throughput side, the first thing you do is to what's called an assay transfer. You transfer the assay into a highly reduced volume, as I already alluded to. So that keeping all the components the same, you're not changing any components. You're starting with what works and you're just reducing the volume. So what you aim for is that you have a good signal at the low volume for detecting the target and how it is altered. We need to use a few parameters to check that it's suitable to go into this high throughput, which means miniaturized format. So that one of the parameters you can check the signal to noise ratio. The signal to noise ratio compares your ability to detect a signal to when there is no signal. So just the noise. And it actually factors in the standard deviation. Standard deviation. If this is foreign to you, no worries. I have a video on here that introduces statistical concept in a really accessible way, so you can have a look at that to see what standard deviation means. Then you have a signal to background. The signal to background basically compares the ability to detect a real signal with the background generated by the components used in the assay. It divides the average signal by the average background. The noise, the buffers, the reagents give their own signal, so you factor that in versus when you've got a real event, a real signal, and so that's what your signal to background adjusts for. Oh, there's also another parameter, a statistic that is used here, and it's the Z prime or the Z factor. Like we said, you start off with an assay that works in a test tube or in a large volume format. You've miniaturized it to 384, 1536 well formats, and then you are going to look at a pilot screen. What's a pilot screen? A pilot screen is just an initial screen where, we mentioned it's going to be like hundreds of thousands of compounds. You can just check a few of those compounds. You can check 5,000, between 5,000 to 10,000 diverse range of compounds to gauge the robustness and hit rate estimate before going to an expensive proper screen. At this point, you only screen at one concentration, just one concentration, but at the pilot stage you actually do it in duplicates just to see how reproducible it is, and then at that point you can actually calculate the statistic that we didn't talk about earlier, which is your Z prime, to really look at your ability to pick a real signal from negative signal. You've done the pilot screen. The pilot screen you did at one concentration. The concentration that's usually used is 10 micromolar. But that all depends on your target and the kind of assay you're doing. In some systems, you may start off at 20 micromolar or maybe more. So you've done the pilot with that concentration. Then we come to what is called the primary screen. This is the screen. This is only done when everything's going well and you're at a good place. So the screen, you select one concentration again, 10 micromolar usually, could be more, could be less, and depending on what you understand about your system, and then you only do one well, singlet, no duplicates. So you really have to, at this, before you get here, make sure that everything is working well. But you don't just assume it's working well. You have some parameters that you can check, so you can put positive controls on the plate. You can put negative control. You will put positive controls, put negative controls, and you can use that to calculate your Z prime or your Z factor. After that, you have to select what you consider are hits. You factor in the Z factor. If it's a good readout, a robust readout, then you can trust what's on the plate that you see as hits flag, because they're like 3 standard deviations away, and you have to confirm them. These are the compounds that have potential, or as we say in this field, putative, potential activity. While you're doing the primary screen, you would have also by now, you should have an idea of another assay. This is very crucial. Another assay that will allow you to weed out false positives. False ones that come up as positives, but they're really not positives. They were interacting with the things you were using, maybe your readout, because usually you're using a reporter, something to report to you that signal is something's interacting with your drug. So there could be components, drugs in there that interact with that reporter and not really with your target, and gives you a false positive, or it interacts, or some of the drugs actually stopped your reporter from reporting, and you think that means something. So you also need another crucial assay in addition to your main experimental assay to see that when compounds are interacting. So that is called a counter screen. So you have the main screen, then a counter screen, and also, of course, it will be you would need what is called an orthogonal assay, and that means like if I used light detection, light emission as my reporter to tell me when a compound is interacting the way that I want, then I can use a different way of detecting that interaction. So if I used a bioluminescent reporter to say the target is interacting with the molecule, then I could use maybe fluorescence as a reporter, and if I still see it in that setting, then I know, yes, it's really, it just confirms it. I just used a different technology to confirm that interaction. So that's an orthogonal assay. Let's say you've done all that, you've confirmed your hits. Then the next step in the high throughput screening process is to determine the potency of the compounds using both the main assay and the counter screen. You're going to determine the potency of the compounds that you've identified. And you do this with a 10-point dose response curve. And this allows you to really, if there's a real interaction, then as you dilute it out or increase it out, starting at a high concentration, as you dilute it out, the signal should go down, down, down. You do it for 10 points, and that means you do a serial dilution of the compound. If you're not familiar with serial dilution, you're in luck. I did an earlier video all building up to this video, and it also gives you an idea of how potent the molecule is. You need that data to be able to draw a curve and determine how potent the drug is. And here the measurement is called an effective concentration 50. You may also be interested not in the effective concentration but the inhibitory concentration 50. So your EC50, IC50 can be measured once you do your 10-point dose response. There's still one step in the high throughput side. Yeah, it's a long process. You've heard of drugs taking 30 years to get to the market, or 30 years or more. Give the compound to be assessed to make sure that the compound that is flagged as potential activity is actually what you think it is. So you do this by sending it to a mass spectrometry lab. They do liquid chromatography mass spectrometry to identify the compound, because sometimes it could be a contaminant of the compound when it was synthesized that's giving you the activity, in which case giving that progressing with that compound and making a drug will not result in the interaction with the target that you had seen. So you have to check via mass spectrometry that the molecule, the compound, is really what it is. You're still not done. You actually have to modify the compound to make it more drug-like. It is highly unlikely that what you find at that point will be ready for use as a drug. Why is that? Well, you know that there'll be, if you're taking this drug, if you're taking it as orally, there are few obstacles the molecule has to go through before it can be available to the target. As the stomach, the pH of the stomach, for instance, highly acidic. It's very, very low pH. It's a pH of 2. So the molecule needs to be soluble at that pH, and not just soluble, but stable at that pH. You have to look at getting rid of side effects. If there's some off-target activity, it interacts with it interacts with your target, that's great, but maybe it also interacts with some other targets in the body. You can't have that, or you can't have it if it's particularly bad. You can reduce that. You can look at reducing its interaction with other targets in the body. Toxicity is an issue. You're going to have to make sure you assess if it's toxic, and you can do that with cell lines, but ultimately these are going to have to be tested in whole organism settings. So first off with animal models, can't avoid that, and then you go into human volunteer trials. And at this point, you're really working with the chemists, the medicinal chemists, to make incremental changes that will allow the molecule to behave in a drug-like way that you can then test in whole organisms, beginning with animals and then human volunteers. Wow, so you can see there are a lot of processes that goes through. So we might look at the Z prime, calculating the Z prime in another video, but for now we can leave it here where we've looked at the process, target identification, target validation, finding an assay to let you see how the target is behaving, and then going on to finding a small molecule that interacts with it, confirming that that molecule really interacts with it, checking the potency of it, then assessing the purity of the molecule first and foremost, and then working with medicinal chemists to ensure that you can make the molecule more drug-like before it can go into whole organisms like animal models and then human volunteer trials. Thank you once again for tuning in. If you have questions, happy to hear them. And yeah, if it was helpful, do give it a thumbs up. Subscribe if you want to hear more of this content, because I definitely think we will look I'll do a separate video on Z prime calculations. And yeah, I'll catch you in the next video. God bless you. Bye.